Hello, everyone, and welcome to Global Summit 2020. And thank you for joining this session where we're going to have a discussion on how to bring long-read sequencing technology to your core lab. My name is Jenny Ekholm, and I am part of the PacBio team. I'm going to start with a short introduction on how to bring smart sequencing into your core lab. And then I'm going to be joined by 4 panelists for a discussion. Each of these panelists are service providers that offer smart sequencing as a service. And some of them represent academic core labs, others are commercial service providers, some of them are from Europe, and some of them are from US. So I welcome you to learn from their experiences and listen to their recommendations. And I would love for you to also join in on the panel discussion, and you can do so by submitting a question into the chat box. My colleague, Jenny Alfrey, will then ask the question in the panel discussion. So the SQL systems are production ready straight out of the box, producing a reliable performance. So as you can see from this chart here on the right, the total yield in gigabases is shown on the y-axis, and on the x-axis you can see the year. And what you can see from this chart is that the total bases are steadily increasing year over year. And this is even when we were affected by, or are affected by, the global pandemic now in 2020. So what can you use a SQL 2 system for? So the HiFi long-read sequencing can be applied to a wide range of applications, species, and sample types. So for de novo sequencing, which is commonly used for building de novo assemblies for plant and animal species, or in the human space for population-specific reference genomes. You can run up to 240 genomes per year at a price point of $1,300 per sample for the smart sequencing cost. Now, if you want to dig a little bit deeper into the genome and detect all variant types, you can do so using the variant detection application. This applies to, currently you can run up to 120 samples per year at a price point of $2,600 per sample. Now, aside from DNA sequencing, you can also do RNA sequencing. You can do whole transcriptome. You can do targeted RNA sequencing, or you can do single-cell RNA sequencing. And you can run up to 240 transcriptomes per year at a price point ranging from $185 to $1,300 per sample, depending on the multiplexing level. Now, for targeted sequencing, you can sequence up to 184,000 samples per year for amplicons at a very affordable cost of $1 to $4 per sample. And for microbial sequencing, there's a quite wide range of sub-applications. You can do de novo assembly, you can do metagenome assembly, you can do metagenomic profiling, or full-length 16S ribosomal RNA sequencing. And you can run up to 57,600 samples per year, depending on the application you choose. And the price point is anywhere from $15 to $1,300, again, depending on the multiplexing level and the complexity of the sample. So how long does it take to recuperate the cost of a SQL 2E system? So when run at full capacity, you can run 240 smart cells per year. And at 60% capacity, which is 12 smart cells per month, and with a markup of 3x, you can recuperate your instrument cost in less than 3 years. Now, you can decrease that time by either increasing your markup or by increasing your utilization. So if you have 100% utilization and a markup of 4x, then you can recuperate the instrument cost in less than a year. So why would you choose a SQL 2E system over any other system, despite the instrument and the applications? It's a very simple answer, and that's because of the HiFi data. So the HiFi reads are not only long, but also highly accurate. And this here is a recent study from Precision FBA where they did a true challenge, comparing different sequencing technologies to one another. And in this challenge, they concluded that the PacBio HiFi sequencing is the most accurate over Illumina short reads and ONT long reads. Now, with the high accuracy and the long reads, it also gives you higher sensitivity to detect all variant types, also variants that are not detected with other technologies. So this here is also from a study or a recent publication in Nature Biogenetics from Lawson and colleagues, where they sequenced the same individual both with Illumina short reads and PacBio long reads. And they found that in the Illumina data, it was missing 47% of the deletions and 78% of the insertions. And this was after applying 11 different variant colors that were specifically designed to detect these variant types in the short read data. So when it comes to a researcher, the cost per base is irrelevant if the data does not contain the answer that they're looking for. So we recently released the Sequel II system, which is now doing the HiFi analysis on instrument. And this really results in a substantial reduction in data compute, transfer, and storage costs. So with the Sequel II system, you get a 90% reduction in file storage needs and a 70% reduction in secondary analysis processing time. Now, as you know, with a project, there is the sequencing cost, but actually the compute cost can be quite substantial as well. So with the Sequel II system now doing the HiFi analysis on instrument, there is substantial compute cost savings. So over a 3-year period, you can actually save over $230,000. So if you are interested in getting started with HiFi sequencing and the Sequel II system, we have now a trade-in and trade-up promotion going on where you can get credit for your old sequencing system, be that Illumina, Nanopore, or an ABI system. Of course, there's some restrictions that apply, but if you're interested in knowing more, please feel free to contact us. And with that, I want to introduce my panelists today. First up, we have Olga Vinner Pettersson from Uppsala University in Sweden. So welcome, Olga.
Hi, guys. Thank you for having me here.
Thank you. With a few words, would you mind describing your services that you offer?
All right, so I'm a project coordinator at the Long Read Sequencing Facility, and we're a part of a huge national genomics infrastructure in Sweden, part of the SciLife Lab. So we are a non-profit, a university-based infrastructure. We are larger than us in Europe is only Sanger Institute. So we have tons of different sequencing machines, have several sequels, and everything else there in between. We were first European users of Sequel I system, and we've been operating with Sequel II since September last year.
Thank you, Olga, and welcome. Let me introduce the next panelist, Charlotte Harris from Corteva AgriScience.
Good morning.
Good morning. With a few words, would you want to describe your sequencing services?
Sure. I am Charlotte Harris, and I am a laboratory supervisor at Corteva AgriScience, where I lead our PacBio team. Now, you may be wondering why an agricultural company would be a sequencing core. We adopted the technology about 5 years ago with the implementation of an RSQ in the lab. We immediately saw a significant benefit in the technology, specifically for our reference program. So we continue to be early adopters of with the launch of the Sequel II. We began collaborating with other researchers at conferences such as PAG, and it sort of just made sense for us to become service providers. So we really specialize in record genome sequencing. We aren't really interested in on smaller projects just because we're also trying to get our own things to the queue as well.
Thank you, Charlotte. Let me introduce our third panelist, Bruce Kingham from University of Delaware. Welcome, Bruce.
Hi, Jenny. Thank you. Good morning, good afternoon, good evening, wherever you are. I am at the University of Delaware. I'm the director of the Sequencing and Genotyping Center. University of Delaware it's a basic research institution. Most of the internal genomics projects are based in agriculture and natural resources, plants, animals, metagenomics, microbial genomics. We also work with investigators from around the world. We purchased our RS system in 2010. Upgraded that to an RS2. We installed a Sequel 1 in 2017. Both of these systems we just recently took out of service. The RS2 was particularly hard to part with because it's such a wonderful system for microbial genomics, and it was our first. We installed our Sequel 2 in 2019, and that system will be shortly upgraded to a Sequel 2E, and we're also preparing for the install of a second Sequel 2E. And we have extensive experience with all of the common PacBio applications, continuous long-read, HiFi, IsoSeq, low-input sequencing, Amplicon, and we also regularly take on special projects which may involve some level of methods development as well. So thank you.
Thank you, Bruce. And the last panelist I want to introduce is Aaron Becker from University of Minnesota. Welcome, Aaron.
Thank you. Thank you very much, Jenny. Yes, my name's Aaron Becker. I work for the University of Minnesota Genomics facility. So we've been involved with PacBio for since about 2017, mid-2017, we got a Sequel system. So we never did actually have an RS2 system. We collaborated with with folks at the Mayo Clinic for that. But we had our Sequel 1 system just recently removed, and we've now had our Sequel 2 installed for approximately 3 months, 4 months now. We've been getting accustomed to the ins and outs of the instrument. We offer most of the standard processes. We do some HiFi reads, we do IsoSeq, and we've done some Amplicon sequencing as well. One of the services that we've been highlighting here for a while at our institution is microbial multiplexing. We seem to have a fair amount of interest in the microbial multiplexing applications. We also have collaborated with our clinical labs on campus to try and develop Amplicon processes in the past to do investigation of long, difficult to sequence, highly variant genes, and with some limited success. And we hope to continue doing more HiFi reads and expanding the knowledge base on HiFi reads. So thank you for having me here today.
Thank you for coming. And with that, I will start the panel discussion. Again, if you have any questions, please submit it in the chat box, and we will bring it into the panel discussion. All right, get the next. There we go. So the first question I have for the panelists is, were there any surprise challenges you faced during the purchasing process? So maybe Bruce, do you have, maybe I can ask this question from you?
Sure. So I wouldn't say that this was necessarily a surprise challenge, but we bought the Sequel 1 in 2017, and it was less than 2 years later that I had to convince the right people to let me spend a few $100,000 more on a new instrument. So that was a bit of a difficult sell. I did that first, and then I had to go back after that and convince them to let me spend even more for the additional computational resources. And this isn't unique to the Sequel 2. It's a challenge all of us face when we're trying to bring in new technologies. But I think for the most part, administration particularly with the rise in core facilities because of the expense with these technologies associated with them. They're not necessarily unfamiliar with it. One specific challenge that we faced is that we were offered some fairly generous discounts on our purchase, but they were contingent on a very short purchasing timeline. So working with procurement at my institution to push this through was a real challenge. They don't necessarily understand the timing. Fortunately, I had all the paperwork in order and a great deal of support at the university. So, the challenges would be those associated with any large dollar purchase for the most part.
Okay, thank you, Bruce. And maybe to hear from more commercial service providers, Charlotte, did you face any surprise challenges during your purchasing process?
Sure. So we didn't really face a lot of surprise challenges on the purchasing side, but anybody who's familiar with the actual RS2 instrument, we found that the installation was a lot more straightforward on the Sequel 2 as compared to the RS2. So the requirements of the RS2 were very strict from an installation standpoint. It used more nitrogen, it was very heavy, it needed to be in a very specific environment. The Sequel 2 is essentially like a refrigerator. It comes on wheels, it's very easy to roll down to the lab. So it was very easy installation. And like Bruce said, I think the purchasing challenges were more convincing the right people that it was going to be beneficial.
All right, thank you. We can move on to the next question. So we know that capital equipment can require, and you already spoke to it, some of you, quite a lot of support from several people or groups on the campus just showing demand. Do you have any advice for people that are going through this, how to navigate the purchasing process on the campus? Aaron, do you have any suggestions?
Sure. Hire a really good finance operations manager, which is what we have actually had here at the University of Minnesota Genomics Center. We have someone who used to be a scientist, but now operates as a finance manager, and she knows the ins and outs of our institutional bureaucracies. So, but barring someone who knows those ins and outs of the bureaucracy, definitely being familiar with the PO process, making sure that you have a good relationship with your finance managers in the finance operations office. Paperwork is always, the bane of our existence, but it's something that we have to do to acquire these instruments. Also, staying in close coordination and close contact with your user base to know what's really the need. Is there an instrument that we don't have that we really do need? So we've used a lot of polling and a lot of surveys to try and make sure that we're staying on top of that. We also have a yearly festival where we invite vendors and our users to come, and vendors can show their wares, but also our users what's sort of the current latest greatest thing. And we usually try to have a theme, and we call this the GenoFest. So that's really been actually quite helpful in keeping us in contact with our user base here on campus, as well as around the upper Midwest, which is where we actually derive a fair amount of our user base from. Other academic institutions around the Midwest. Yeah.
Thank you. That was a lot of great advice. Bruce, do you have anything to add?
Sure. So kind of building on what Aaron had mentioned, the first and most important is to know the purchasing process at your institution. Each has their own idiosyncrasies. Who needs to approve? Is there a bidding process? What documents do they require? What's the timeline? And the faculty are really the drivers for academic research. So if you can get an influential group of them on board, your job with administration is going to be much easier. And it's helpful to put some thoughtful effort into a business model that describes how the purchase will be a success story, and not just for your core, but for the institution as a whole. And faculty or administration is really now starting to better understand how core facilities, the impact that they have on faculty recruiting, as well as promotions to tenure for faculty, bragging rights for the institutions. Administrations like to be able to tell their colleagues that we have these types of technologies, peer-reviewed publications, and the positive impact on grant proposals as well. And it's important, I think a lot of us in these types of roles, we tend to undersell ourselves. This work is really hard. It's not easy to do. It's a very high level of science. So it's important to convince administration that they need us. They need cores. They need technology centers centralized to draw in the talent that'll write multi-million-dollar grants. And remember that administration and faculty need us as much as we need them.
Yeah, that's really a lot of great advice. Thank you, Bruce and Erin. So I discussed a little bit on terms of—
Hey, Jenny Ekholm. Really, really quick, I just want to say there are a ton of really awesome questions.
Great.
And I do think we're going to address some of them as we move forward, but I just want to reassure the audience that we will absolutely follow up with you guys afterwards if we don't get to your question. Some really good ones. So let's wait a second because I think we're going to get to some of them.
Thank you, Jenny. Yes, so I touched upon this in the beginning a little bit about recuperating your costs for your instruments. Describe how that kind of fits into your Sequel II plan. Let me see. Charlotte, would you mind taking this question?
Yep. So the biggest benefit that we have found for throughput is really just the greater yields that we're getting from the Sequel II. So again, if you think back to the RS2 or even the Sequel, it would have been really hard for us to recuperate those costs coming from the commercial side because the projects may not be large enough for us to generate the income to pay for the instrument. So again, throughput has been a huge win for us. We're really happy with the throughput improvements that we've seen in Sequel II and that we anticipate we're going to continue to see. And it's allowed us to take on these much larger, more complex projects and really get a better profit margin.
Thank you, Charlotte. Erin, do you have anything to add?
Sure, yeah. So the university here, so we work as an internal service organization. So we have to recover our costs, of course, for these instruments. We have very little in the way of subsidy, if any subsidy at all, actually, for our operations. So that brings into the whole consideration of trying to figure out what exactly are your costs. Figuring out time on instrument, actual cost of instruments. So I find that it's sometimes difficult to come up with labor costs and instrumentation costs that are really accurate. So, we expect that we're going to be operating potentially at a little bit of a loss here, but for the first year or two. But we expect the usage of this instrument to ramp up. I don't think people have really grasped what HiFi Reads can do for them yet. Maybe this is somewhat due to the pandemic, but we're hoping that our microbial multiplexing service can help us sort of expand our role here in the Midwest and worldwide. But yeah, it's definitely something we're going to have to continue working on to make sure that our throughput continues to stay active. Because that's been somewhat difficult with the pandemic, making sure that we're getting enough information out there about, hey, we've now got this system. Let's start using it. So that's been a bit of a challenge.
Yeah, 2020 has definitely been a special year. Yeah. And next question is that I have for the panelists is, how how has adding a SQL2 system to your core services changed your business? And I'm going to ask this from Olga, and apologies, she has not been ignored. It's just they have a very different purchasing system in Sweden. But maybe you can start with this question.
Yes. So what I can say from our part, so first of all, speaking of business, we have no business, we're non-profit. So the Swedish academic users, they're just paying for the reagent cost, and the instrument depreciation and so on is taken by the Swedish taxpayers via the governmental and other funds. But speaking about number of projects that are coming in. Since we became a certified service providers and we made the world aware that we have the SQL2 system, the amount of project requests that I'm getting from abroad, it's just went uphill like that. So we're getting lots of requests, and the queue on the machine is actually growing steadily. So we need another machine. Yes, but we see a great interest in the HiFi project. So HiFi is an amazing application. We see a huge demand on that from many different fields of biology, biodiversity, biomedicine, and everything else possible. So yes, business is good. Business is good.
Bruce, do you have anything to add?
Sure. So during our 10 years running these systems, with every incremental improvement, whether they were hardware, software, or chemistry, we've seen that door open up a little bit wider, and a few more investigators walk through that door and start using the technology. So you could imagine over the course of 10 years, along the lines of what Olga had mentioned, a lot of the inquiries are overwhelming at this point with regards to numbers. It's increased the number of investigators we work with. It's really broadened the reach. 10, 20 years ago, most of us that ran cores were largely working only with internal investigators. And there's some understandable reasons for that. 10, 20 years ago, these technologies were much less expensive, sequencing technologies in general. But as the cost of these technologies increased, and the requirement, at least in the States, for core services to basically do their best to support their operations, it's required us to work with investigators outside of our respective institutions. With that, we work with many more groups, and it's international in scope. Further, the way that it's changed the business, more so on the internal laboratory side, it's really expanded the skills and the expertise of my team. The skill set needed for long read sequencing is very different than that for short read sequencing. The era of PCR really took a lot of those skills out of the genomics field. Some of those differences are minor, some are major, and most are connected to working with high molecular weight DNA, which was a lost art. But now a lot of scientists are regaining those skills. Further, it's important to set expectations with investigators because this is not short read sequencing. There is not the same level of certainty, particularly with genome assemblies. We're sequencing the native DNA, and we know that based on the species we're working with, or details of the samples, whether they be the origins, there's a lot of challenges associated with them, and how we can work with that DNA and be able to sequence it. It's not like short read sequencing where the library preparation is really standardized. You're doing amplifications. When we're working with different types of organisms, there are a lot of different compounds that can coalute with the DNA, may be bound to the DNA, and there are lots of different tricks on how to remove those, how to deal with those, so that we can effectively sequence it. But there are guidelines. We have guidelines on expected yields. But as we all know, working with PacBio, that can range widely. Yeah. It's been important for us to expectations with investigators that we're working with.
Thank you, Bruce. So next question, how easy has it been to implement the Sequel II system to your core? So if you could give some examples of challenge and success that you have faced, Olga?
Yes, actually, I would like to pick up on what Bruce said right now. So, and also I've seen that in the chat, people are asking very similar question. So even moreover, I can say that here every project is unique. There is no such thing as consequent yield from a single smart cell. So there is no such thing like several samples from the same sample, perhaps same organisms performing in a similar way. It's just, it doesn't happen. So even though you have human biopsies of human cells extracted from the same lab, very often you see discrepancies when you load them onto the machine. So that's one thing that we learned when operating Sequel II system, because we had PacBio instruments since 2013, so we know the challenge, right? So if before on RS2 or on Sequel I, what we would do, we'll make a library. We'll try to make all possible DNA QC. And very often DNA QC will tell you just that big about the sequenceability of the sample, but it will not give you a 100% answer. So the best is actually to put the sample on the machine and check how much data you're going to get from there and what is your read depth and the length and so on and so forth. So with RS or with a Sequel I, it was easy. So you put one smart cell and then you can titrate different on-plate concentrations and so on. With Sequel II, one smart cell perhaps is all that you need. Or one smart cell is actually quite expensive, $1,300 or alike. So in that sense, it's actually very difficult to a priori tell you how you're going, I mean, is it going to perform? So is one smart cell enough to sequence a bad genome? Is two smart cells enough to sequence a mammalian genome? The answer very often is no, it's not. So that's from the downside of having Sequel II system. There is no good parameter to estimate the quality of the material and the library before you actually sequence, and there comes the cost, right? The second thing that I would like to point out, that the entire lab logistics had to change because the throughput of the machine is much lower. So that's in terms of smart cells that you can process a week. So before we could easily do 34 smart cells in our Sequel I machines, right, a week. But now we can do only 5. And that means that if we have projects when we have large genomes, you make 1 or 2 libraries and then you just run them. Of course, you're getting more data per run, so then you sort of be done with sequencing much faster. But if you have a project with 20 samples, for instance, like ISO-seq samples or, I don't know, small genome samples, then you actually have to really plan very tightly. How do you hear me? Guys, do you hear me?
We hear you now. Yeah.
Yes, good. Because I disappear from the screen. Yes. So you need really to figure out when you make library, when you bind your polymerase, when you put it on the machine, because the bound libraries, they have very limited time span, right? But on the pro side, that's a really cool type of data. So we've been reached by people who are working again with very large plant genomes where they try to assemble that with any other possible technologies. And only with HiFi that you get from the machine, they could actually start looking at the variation in the genome, look at the gene family expansions into the duplications. into the duplications, into the overall genome structure. So, but again, another good thing, actually, I must say, the Sequel II system is the first PacBio system that was working from the shelf. So we installed the machine, and since day one, basically, we had no problem with the machine itself. We just learned how to fine-tune it, how to make our libraries work. But with the machine itself, no problem. With Sequel I, it took us a year to come from the test to the production. So PacBio, thumbs up, good machine.
We'll listen to your feedback, and we try to make things easier. That's our job. Aaron, do you have anything to add?
Sure, yeah, I'd love to. I will echo the Sequel I versus Sequel II differences that Olga was mentioning there. That we definitely found out of the box, the Sequel II has been very good for us. There really have been very few technical challenges from the point of getting the machine up and running. So it's actually been quite easy with the Sequel II. And I'm very happy to have that happen at this point in time, because we've been with the way we've been scheduling folks at this point in time. It's nice to have something that was installed during the pandemic, and it worked right out of the box, and we're able to actually keep our business going. So it was one of the worries for us that we were going to fall behind at times if we didn't have the Sequel II. That was one of our biggest considerations, that we had Sequel I for a while, and we knew that the Sequel II's capacity was really going to be important for us as an institution to have. So getting this instrument in and having it work right out of the box has been terrific. So from a sample perspective, we do a lot of work with plant genomes and the agricultural groups here on campus, as well as doing a fair amount of microbial multiplexing. So our experience with these variations in performance of these libraries is pretty substantial. As Olga said, it's very difficult to do QC without actually running the darn library, and that will tell you a lot about it is just by actually collecting the data. So there's no PCR method that allows you to assess the quality, and that's again, it's just par for the course. It's the way the technology works right now, but. The biggest challenge then, aside from the sample quality, it has been for us is our informatics side. We don't have a 2E, and we're not going to be getting a 2E, I don't think, anytime soon. So we're going to be probably struggling with the infrastructure and analysis side of demultiplexing HiFi reads, doing a lot of high output Sequel II cells that can actually really tax our infrastructure, even at the Supercomputing Institute. So we have some work to do there. So advice to any groups that are considering getting the Sequel II instrument, that your informatics challenges could be severe if you are not prepared for that.
Absolutely. I second that.
Yeah.
We had to write a separate grant for the compute infrastructure outside of the cost of the machine. So yes, it's a very valid point.
Thank you. Now I have a question that I was hoping to get an answer for all of you. So what kind of demand are you seeing for the HiFi reads? So Charlotte?
Sure. So we really only run HiFi sequencing. So I would say in our area, the demand is quite high. From an agricultural perspective, HiFi reads are really the only thing that allow us to assemble large and complex genomes. Example of this is oat. And this was previously not feasible. So we have been early HiFi adopters, and the oat genome, which was a collaboration with PepsiCo, was definitely really enabled by HiFi. So it's been very impactful for us, and it's, again, it's really the only thing that we're running in our lab.
Thank you. Olga, I already saw you doing this, so.
Yes, I mean, HiFi are amazing for resolving structure variants as well as the CLR. HiFi are, and the throughput of the machine, they are beautiful. Even if ISOSeq is not really run in HiFi mode, still the quality of those ISOSeq reads, it's amazing. They are amplicons, like large amplicons for weird minor variant detection. I mean, huge demand, huge.
Thank you. Aaron?
Yeah, it's slowly ramping up here. Once we got the word out that the instrument was installed, like I said, it's really been since midsummer that we've really been up and running and had this this going. But yeah, the demand has been ramping up. Clients who used to had considered potentially doing PacBio projects in the past, and realized they had to do maybe 8 cells to do one genome. I don't think that there's been enough of a marketing by us as a genomics institute potentially to get people to understand what the difference could be, right? You could do now this on 1 to 2 cells maybe, where it used to take you 8 cells or more, and the cost differential. So that's partly on PacBio. It's partly on us to get the word out and make sure that people understand the advantages. But yeah, it's definitely coming up. So a couple of projects that only last year would have cost us, $12,000 for a client to do, we might be able to do it for, a third of that cost or less.
Thank you. And Bruce?
Yeah, so all HiFi all the time, pretty much. A little bit of ISO-seq, low input, and amplicon sequencing mixed in. But yeah, it's that technology or that application is a huge advancement. The major advantage for us in the laboratory is that the requirement for the fragment sizing of the DNA is not as important. We don't necessarily need that 30, 40, 50 kb fragment size. The downside to it, to a certain extent, is the input requirements for it. But being in agriculture, most of those projects, we have a sufficient supply of material and tissue to extract DNA from. The challenge are those projects where the sample and the tissue is more limited.
Okay.
But then the low input protocols offer us some alternatives for those, which have been very effective as well.
Thank you. So Aaron touched a little bit about on this already, but how are you marketing your services, Charlotte?
So to be completely honest, we really haven't done much marketing. We've been pretty lucky. We met people through conferences, and we've really worked on building collaborations with academia. And we have quite a few ongoing collaborations from PAG 2020 that are just continuing to grow. So really large conferences such as PAG have been a key for us for marketing.
Thank you. Bruce?
Yeah, so I'm occasionally lightly pressured by one individual at PacBio about tweeting more often, and I won't mention Wendy's name, but that's not my wheelhouse. I wish it was. I try to do it. For our laboratory, it's mostly word of mouth. We've been in this business a long time. From the very beginning, so my laboratory has that advantage, and for the most part, beyond that, it's in-person social networking at events. We have very close relationship with our PacBio team, and that's led to involvement in SMART grants, hosting a UGM. The company PacBio has been very beneficial and helpful with helping to sponsor seminars and events at my institution. I've found that, at least internally, that's what really drives a considerable amount of interest towards these projects, is when they can attend these seminars in person, speak face to face with the PacBio representatives. Certainly this is an era of social networking and technology, but I still prefer face to face. And in my experience, a lot of the investigators that we work with tend to respond in a more positive way to that as well. And every year the technology is more effective at selling itself as well. So kudos to PacBio, because we have to put less effort into marketing our services.
Exactly. Data sell themselves.
Mm-hmm.
Thank you, Bruce and Olga and Aaron. So describe your customer base. Do you get a lot of new customers, or are they repeat customers? Aaron.
Yeah, our customers, we have a few sort of core users for our PacBio technology. So, probably 20% is repeat, and a lot of the business we've gotten over the last 3 to 4 years has been more new users who are coming in and first time using the technology. A lot of what we've done that's repeat has been either iso-seq or microbial multiplexing for a couple of users. That's where we're finding that. But you'll see that the new users are those people who have a genome or two that they want to do. They get their data and then you don't see them again. So I think that at least we haven't. Maybe the HiFi Reads and the sequel 2 are going to change that for us. So that's the hope. That's the reason for having this, to get more new users realizing the advantage of the platform.
And Olga?
Right. So we have both. Speaking about older users, they're coming with more interesting questions, like lots of work on repeat expansions, the centromere structure. Those are mainly evolutionary biologists who really would like to go beyond SNPs and look for the structural variants for population studies and evolution of complex traits. And speaking about new, we are more and more getting people who are doing structural variations in humans or disease-causative mutations, because they've done exome sequencing, found nothing, done Illumina, found nothing, done 10x, found nothing. And then they're coming for PacBio, and sometimes they're finding some interesting things. And also Sweden, the SciLife Lab, my infrastructure, we joined the Earth by Genome project. Nice. And then there is a huge interest in de novo genome assembly from that community. So we're getting more and more requests from people who done their work using a bit lower quality references. Mm-hmm. So there is a huge inflow of interest from that community as well.
Okay, thank you. So we talked about the sequel to E having the on-instrument HiFi Reads. How would this help your core, Charlotte?
So this is something that's to be determined for us. We are lucky enough that we have a large cloud infrastructure. So, this is not really impactful for us right now. We are going to be doing a cost analysis to compare what the better option will be. But for a smaller core that has less computational support, this would absolutely be a critical component for them.
Thank you. Aaron?
Yeah, I think I've probably mentioned this already a bit. That we definitely have a need to get the word out about what HiFi Reads can do, and this is going to help us continue to expand, our technology offerings. And being that we're a little bit behind in getting the instrument installed, I think that's our challenge going forward here. But it's definitely going to help our visibility, I think, in the Upper Midwest with our ag clients, as well as those folks who are coming from universities whose cores just don't have maybe the flexibility that we do to offer these latest technologies. There definitely are challenges at American universities right now in funding some of these items, right? And because of having to recover costs, so we believe that it's definitely going to make us a known commodity here, at least in the Upper Midwest.
So what unique benefit does the Sequel II system bring to your core lab and that is not serviced by other technologies? So Olga?
Right. So main benefit from the Sequel II system is HiFi Reads. That's Illumina quality reads up to 25 kb. That means that the genome sequencing is cheaper and you do not need to have a polishing. So with other long read technologies, we have Promethean, we have everything. So there you still need polishing. So in that sense, and then you also need higher coverage. So that's the reason why the DNA is sufficient. we would always prefer to go for PacBio just because it's so much easier to work for that. Bioinformatics off the shelf, the reads of higher quality, no need for additional polishing, albeit from perhaps, I don't know, high C on top of that. That is an Illumina, well, structural variations, we all heard Jason's talk, not Jason's, Ivan's talk, and some other things like, isoform discovery and single cell isoform discovery. That is something unique for the system.
Okay. And Charlotte?
Yeah, like Olga said, really the HiFi reads are such a unique benefit, and they're a critical component for us for our multi-data strategy for generating reference genomes. The other thing that I would like to point out is that PacBio does have a reliable patient supply. We have not always seen that with different long-read applications. And I mean, having multiple instruments that are constantly running with very, very little downtime is extremely important to us, that we can reliably get reagents when we need them.
Thank you. So just to all of you, would you recommend a sequel to E? Probably some people here online that are trying to decide, should they do this, should they not? What would your recommendations be and why? Bruce?
Of course, it's the platinum standard for long-read sequencing. And I would say for sequencing in general. I may be a bit biased there just because of my relationship, but undoubtedly, yes. It's going to be more important moving forward as well.
Yeah, agree with all that. Yeah, for sure. If you can swing it, if you can corral the funds and get your institution to understand the importance of this sort of next wave of NGS technology, I think it's very important. The HiFi reads and the system are definitely going to be something that you want to get.
Olga?
If 2E was as good off-shelf as Sequel 2, definitely go for it, especially thinking about the amount of money you need to invest in compute infrastructure. By having just 2, definitely go for E directly.
Charlotte?
Absolutely. I mean, there's really no other data type like HiFi, and we've seen such a significant reduction to our genome pricing. It's just been a huge win for us. More and more resequencing applications and RNA applications are moving to Sequel 2, and I suspect it's going to only continue in the future.
Thank you. And Jenny, Alfred, do you have any questions from the audience that have not been addressed?
Absolutely. Can you guys hear me?
Yes.
Okay, great. So there were a couple of technical questions about shearing and that we will follow up with FAS, just very specific ones. But one is about support. So the question is, what happens if we need support from PacBio? Is this easy to get?
I can answer.
I don't know.
We've been working with Applied Biosystems, Life Sciences, Thermo Fisher, all other companies that should not be named, and even other companies that should not be named. PacBio provides great service. PacBio field application specialists, if they cannot find the answer, they'll go to the research and development team. The service engineer that we're having they are amazing. Community is amazing. Seriously, PacBio is like one big family. I'm not paid by them. I mean, we had, since 2013, it was a very nice experience working with this particular company. So you have all the help from them in running your core facility in all possible levels, not just with the machine, with auxiliary equipment, how to make your libraries, how to share, and so on.
Anyone else?
Yeah, I agree. The company's grown over the past 10 years, but it still has the feel of a small company. We've always had very close relationships with our reps, whether they're the engineers or the application scientists or the sales reps. Much closer relationships than with other vendors.
Mm-hmm. Yeah, the grant programs have been good for us as well, as far as ways to get the support and get the word out about the services. So yeah, we've definitely appreciated the FAS and the sales support on that side.
You want one last question, Jenny?
Yeah.
So this question is, this is interesting, but we're not sure if there's demand on our particular campus. Is there anything that PacBio can do to help us see how much demand, or at least help spread the word locally?
Is this question for me or for the panelists?
I guess, yeah, for PacBio. Jenny, I would answer that and then see maybe if anybody else has any feedback.
Yeah, so we definitely have different strategies that we can go about. So as you heard, it's very important for us that you as a customer get up and running right away. And we do, we have a great engineering team, a great field application team that just will help and support, make sure the instrument runs smoothly and that you are ready to prep. And if you have any questions, we will answer them. We also have other additional programs. So if you're a certified service provider, you get additional support. We help you update. Actually, this is actually for service providers now available. We help you to update your web page. So we have a portal where you have all imagery available to you. You even have text that you can add to your web page, just because we know that not everyone has, dedicated personnel to do this. So we want to help you as much as we can. And then your local team will work with you and see what fits your needs the best. So as you already heard, we have these so-called PacBio days where we invite, you can invite people from your institution or nearby institutions, and we will help you have speakers coming up, talk about the technology, other users presenting their results, and just trying to generate demand. We also have different types of other activities. Some we have the corporate smart grants, which you have to be a certified service provider to be part of. Basically, we have on different topics, and we open it to the entire world, and you can submit an abstract to apply for the smart grant to do a project. And that, of course, also brings in a lot of additional demand for whoever is the dedicated CSP for that smart grant. But we also have the local smart grants where we do similar things locally for your region. And please, I invite the panelists to add. I'm sure we do try to cater to each customer, listening to their needs, and we come up with something different for each one of them. But if anyone else has something to add, please do so. Quite
sure. Didn't want to. Yeah, sure. I can add something. So again, like I said before, so we've used this curated user list that we have. So I don't know if a lot of cores are able to do this, but most of them have an idea of who their sort of regular user base is. Whether or not they need long read technology, I guess that's a question that you need to put to them. Is there a need for it? We have this ongoing relationship with our user base, and we host small conferences, and we host small gatherings that promote use and themes in biology that might be useful to researchers. So that's definitely helped us keep in contact with what the latest demand might be. Making those theme-based operations or theme-based settings can give you a real sense of where your user base is.